What is DNA? DNA stands for deoxyribonucleic acid. It is the material that contains the information that determines inherited characteristics. What is the structure of DNA? DNA is the shape of a double helix. DNA is sometimes compared to a spiral staircase. The structure of DNA is important because it helps us determine how it replicates, stores, and transfers genetic information. There are three main components of DNA. The first component of DNA is the nitrogenous base. Then there is the sugar, which is also known as deoxyribose. And last but not least is the phosphate group. A nucleotide is a subunit of nucleic acid chain, consists of sugar, phosphate, and nitrogenous base. Nitrogenous bases. Nitrogenous bases are complementary to each other. There are 4 different nitrogenous bases that are held together by hydrogen bonds. The first nitrogenous base is adenine. Then there is guanine. Then there is thymine. And last but not least is cytosine. Adenine is paired with thymine. It is held together by 2 hydrogen bonds. Guanine is paired with cytosine and is held together by 3 hydrogen bonds. When paired together, the nitrogenous bases fit together like puzzle pieces. Importance of complementary base pairing. Helps hold the 2 strands of the DNA molecule together. It also helps explain how DNA replicates before a cell divides. There are many items involved in DNA replication. One of these items is a DNA helicase, which is an enzyme that separates the DNA strands. The replication fork is a Y-shaped point that results when the 2 strands of a DNA double helix separate so that the DNA molecule can be replicated. DNA polymerase, an enzyme that catalyzes the formation of the DNA molecule, or enzyme that ends complementary base pairs to single-stranded DNA. DNA ligase is an enzyme that joins the gaps. Semi-conservative replication is when in each DNA double helix, 1 strand is from the original molecule and 1 strand is new. In step 1 of DNA replication, the DNA helicase breaks the hydrogen bonds between the nucleotides. In step 2 of DNA replication, the DNA polymerase adds complementary nucleotides to single-stranded DNA at the replication fork. In step 3 of DNA replication, the DNA ligase forms covalent bonds between deoxyribose and phosphate.